I want to start out by saying this is work that was done by obviously a lot of different people, just as we've had with all the other talks. Eric Hazelton has done an awful lot of the work. He's a PhD student of Karin Kettenrings at Utah State University, and some of this work is also mine. Dennis Wiggum at CERC is also, he is the lead of the Phragmites project, and so he was not able to come today, so I'm here instead. But obviously, most of you have been out and seen the wetlands, nice native diverse wetland that suddenly becomes a sea of Phragmites with exactly one plant species. And it just about, one of the things that we have found is that very, very early in invasion, those native species are almost entirely excluded. So when Phragmites comes in, it isn't like it comes in a little bit and you still have some native species coexisting with it. No. Phragmites comes in and it very quickly establishes a monoculture that then expands and pushes out all of the native species. So that's something that happens very early in invasion. But later on in invasion, Phragmites starts to build up the marsh surface. And that's where we start to become concerned about some of the greater ecosystem impacts beyond the biodiversity of the marsh vegetation itself. And so what we see is that this buildup of marsh surface and vegetation starts to encroach on tidal creeks. It can actually totally close in tidal creeks, and it can expand out into open water. Now, I know I'm sort of preaching to the choir here in terms of the effect of Phragmites, but this is an example of a meta-analysis that was done by Kim Dibble a couple years ago, and what she did was to look at the effect of a Phragmites marsh versus a nearby native marsh on nekton and invertebrate taxa, and swimming invertebrates are included within the nekton, whereas the invertebrates that they focused on were more sort of within the substrate and on the marsh surface itself. And what you can see here is that pretty much all of these have effects to the left of this zero line. So whether you're looking at nektons or invertebrates, whether you're looking at juveniles or adults, and regardless of sort of what metric you're using for condition, for diversity, for quantity of the taxa you're looking at, that pretty much all of these things show a negative effect of Phragmites marsh on the other organisms in the system. And so this gives us a really strong reason to be concerned about what is happening when Phragmites is invading these marshes. So the problem. The non-native form of Phragmites has had a massive expansion in the Chesapeake Bay tidal wetlands since the 1980s. Basically what we found is we've done a lot of research in understanding how this invasion happens. So what happens is you start out with Phragmites with rare long-distance dispersal. You have a nice subestuary with a lot of nice tidal marshes in it, and every now and then a seed of Phragmites manages to wash in and become established in a marsh. Well, what happens is these initial patches often don't expand very quickly. They kind of hang out. They look like they're pretty innocuous. They don't really seem to be doing anything. They don't promote additional spread, so you don't see a bunch of other patches popping up. And what's going on here is you have a patch that's made up of a single genotype of Phragmites. And what happens is the seeds that it's producing are almost entirely inviable. So these seeds are not going to germinate. They're not feeling any additional spread. But over time, eventually, other patches establish close enough to the first one, or even right next to the first one, so that or patches grow together, so you start to get a lot of viable seeds produced. Now, Phragmites needs different genotypes in order to produce seeds. So pretty much once you've got two genotypes there, they can exchange pollen, and that's when you start to get lots of seeds produced, and that's when you start to see the invasion explode. And we've been able to track this and looking at patterns of genetic diversity in different subestuaries of the Chesapeake Bay to show that this really does seem to be what has happened here. And once you get all of these seeds produced by a nice diverse patch, these nice viable seeds, they disperse out some distance from the original source, and they colonize either disturbances within wetlands or even areas that are not currently disturbed. Now, the initial establishment of Phragmites seems to largely be associated with large disturbances. But once you start to get all these seeds produced, you have so many seeds coming in everywhere that it doesn't even necessarily take a disturbance to allow Phragmites to establish once you've gotten to this point where there are lots and lots of seeds available in a subestuary. However, even though the seeds can establish in these undisturbed areas, the establishment of new patches still seems to be fueled by development, either at or near the shoreline, and eutrophication, especially nitrogen enrichment. And so what we see is Phragmites has sort of a very vulnerable stage early on when it's in seed germination, where little tiny Phragmites plants are very, very susceptible to being flooded and to being dislodged. Once they grow to a certain size, they're pretty impervious to that. And what happens is if you have a nitrogen-rich environment, they get very quickly from that vulnerable stage to where they're suddenly pretty invulnerable. So what we've been trying to understand as part of this project is how the establishment of new patches is fueled by development. And what we can do is we can say, all right, if you have multiple genotypes in a patch, you know that means multiple seeds established there. And so what you can do is you can use patterns of genetic diversity to understand what has happened in terms of seed establishment, to understand what conditions promote establishment of new Phragmites patches. And I'm going to show you just one example of that. One of the things that we were interested in is the extent to which shoreline hardening is promoting establishment of Phragmites, because oftentimes you see a patch of Phragmites associated with a piece of hardened shoreline. And so what we did was we went out to 9 different subestuaries. You can see there's 3 here that were primarily developed, 3 here that are primarily forested, and 3 over here that were primarily agricultural in terms of the watershed use. And then within each of those different primary watershed land use types, we found patches of shoreline that had, they were either natural, hardened by riprap, or hardened with bulkheads, and found Phragmites patches associated with those different shoreline types, and looked at how diverse they are. And said, all right, well, if they're more diverse, that means more seeds have established there, and that suggests that that type of shoreline might be promoting establishment of Phragmites seeds. So we have here in the green forested subestuaries, in yellow agricultural, in the red developed. And what you can see, this is just a number of genotypes out of 10 samples. So what you can see is that the natural shorelines in primarily forested watersheds have, on average, just over 1 genotype each out of those 10 samples, which tells you a lot of those patches have only a single genotype. They're not producing viable seeds. They're not contributing to additional spread. Whereas if we go up to, even within forested subestuaries, if we go up to pieces of Phragmites patches that are associated with bulkheads, they are somewhat over 2 genotypes, have over 2 genotypes on average, suggesting that considerably more of these now are producing more seeds that then fuel additional spread. And we see pretty much the same pattern, whether if we're looking at primarily developed subestuaries or patches in primarily agricultural subestuaries. You can also see, obviously, that there's a big difference between patches that are in forested compared to agricultural and developed subestuaries. And that, we think, it has to do with the effect of elevated nutrient levels in the waters, promoting very rapid growth of Phragmites, and so supporting better establishment of seeds. So we've got sort of 2 factors going on here. Certainly the landshed water use affects the spread of Phragmites because it affects the ability of seeds to establish. We also know that in watersheds that have lots of development in agriculture, those that have elevated levels of nitrogen in the water, Phragmites produce more seeds in those watersheds. So not only are there more seeds coming out, there are also more establishing in those subestuaries. So this is the type of analysis we did. One thing that was pointed out to me was that, well, a lot of these bulkheads have been around for a lot longer because we don't really do those things anymore. But we followed this up with a study of just newly establishing patches associated with unhardened riprap or bulkhead shoreline. And what we find is that this exact same pattern is established right at the beginning of patch establishment. So that sort of tells us that these different types of ways of hardening the shoreline are promoting establishment of Phragmites seeds compared to what would be found in unhardened shorelines. So this is the type of analysis that we've been doing. And we've also been looking at sort of how Phragmites is spreading and where. So to give you an idea here, if we look at, for example, the Road River, because of course that's near and dear to our heart at Cirque, we had in 1970, there were 5 patches. There's right here, 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 and here. 5 little patches of Phragmites that had probably been there for quite a while, not doing a whole lot. Well, when we went back and looked at the same watershed in 2007, you can see all of the yellow covering this, and we identified 212 individual patches of Phragmites. And this is by no means atypical of what's been happening throughout the Chesapeake Bay in a lot of different subestuaries. So this is what what we're focusing our work on is sort of how does it get to this point, and what do we do about it? So the current state, we've used the same studies that I was telling you about with looking at patterns of genetic relatedness, and we've been able to use those to demonstrate how far Phragmites seeds go. And this sort of matters for if you're trying to manage portions of an area and remove Phragmites in portions of an area. How big of an area do you have to worry about? And what we found was that most effective seed dispersal is within 100 meters of the source. So most seeds are staying pretty close to home. There is significant dispersal out to about 500 meters. And beyond that, it's really pretty rare. So this tells you that if you're trying to manage a single patch of Phragmites and you've got another patch right next door, you are going to be in trouble because you're going to have a constant seed rain coming in from that patch next door. Whereas if you are much farther away from your next Phragmites patch that actually happens to be producing seed, you're going to have a lot more success. This also is important for if you're, say, putting in a living shoreline that happens to be right next door to a great big Phragmites patch. Now, I didn't mention it when I showed the slide with the unhardened riprap and bulkheaded shoreline. Some of those riprap patches are actually living shorelines. But from the perspective of genetic diversity, the patches associated with living shorelines are no different from those patches associated with a riprap revetment that's done in a more standard way. If Phragmites gets established in there, it will take off and it will get established well. There were, I want to point out, an awful lot of living shorelines that had no Phragmites in them, though. So it's not that it doesn't ever work, it's just that if you let Phragmites go and you let Phragmites colonize, it will take over just as if it were a regular riprap shoreline. So, we also found that wetlands in a lot of subestuaries in Chesapeake Bay already have so much Phragmites that control and management may just not be practical. For example, this largely agricultural subestuary here, virtually all of the wetlands in this subestuary were almost 100% Phragmites. But, and nearly all subestuaries in the Chesapeake Bay have at least some of the invasive form of Phragmites, but some have few enough patches that control is probably still possible. So you've got a patch here. This is Parker's Creek watershed, and it has very, very few patches. They are actively trying to remove Phragmites in those patches. But without management, almost all native wetlands will be dominated by the invasive form of Phragmites, and there's currently no tool to support effective management. So we went out and did a 6-year removal as part of this NOAA project. We went out to same 9 watersheds that I told you about before. We identified pairs of patches of Phragmites and a patch of adjacent native marsh. We went out and treated one of the stands in each watershed with glyphosate for 3 years, helicopter spray the first year, backpack spray follow-up the next 2. All the sites we found had a native seed bank, had a significant seed bank associated with native species, suggesting recovery to be possible. After 6 years, some of those sites had recovered with native plants, others had not. But if you look at sort of an ordination of those plant communities, you have the native marshes here, Phragmites marshes that we haven't done anything to here. Those were removed Phragmites. They're different from the 100% Phragmites communities, but they don't approach the native marsh communities yet. The recovered differed among subestuaries, and this was probably a function of soil characteristics, and I'll follow up on that in just a second. But recovery also took longer than we expected, with some sites taking over 4 years before there was really any recruitment of native plants. And of course, Phragmites is recovering really fast, and in some cases much faster than the native vegetation is, suggesting that it's going to take additional years of management to keep that going. Now the 3 examples I want to show you. One is a fairly good recovery. This is the Patapsco River near Baltimore. It's got annuals in it, but it also has some perennials such as Shanoplectus and Spartina. You can see there's still Phragmites here in the back. This is a relatively solid marsh, nice sandy underfooting. The marsh itself has pretty good integrity even without the Phragmites there. In contrast to that, on the Severn River near Annapolis, there was very poor recovery. Really just annuals, really only Plukia, and a really rapid return of Phragmites. So this was a marsh that was relatively deeply inundated, relatively unconsolidated substrate, does not hold together well when you remove Phragmites. This might be an example of something where we would have to plant in native vegetation to give it a head start before that marsh starts to go away. Similar example on the St. Leonard River near Solomon's Island. We had some recovery, some Acnita, some Spartina, but it also took 4 years before the recruitment began. And there was a lot of peat loss, a lot of marsh subsidence, and Phragmites is going to return. It's returning with a vengeance. You can already see it here, and it's going to return completely without additional treatment. So that suggests there are some places that maybe Phragmites management is more likely to be successful than others. So what this means for management is that it needs to occur on a large enough scale to minimize recolonization through seed dispersal. Removal of Phragmites, just one patch at a time, just isn't going to work very well. In most instances, this means that the management's going to have to be at the scale of the whole subestuary, or a subestuary, and that's a difficult sell in a lot of cases. But we need to identify subestuaries that still have high-quality marshes with very few Phragmites patches, and these are the ones where restoration of these systems is much more likely to be successful. We're also working very hard to develop management tools that could be used to guide landowners and managers as they address the scale of this problem, and to develop guidelines for the effective removal of Phragmites and restoration of native vegetation. So identify those wetlands where it's more likely to be successful and those where it's less likely to be successful. And I would say also a public program is needed to alert citizens, resource managers, and politicians to the scale of the problem and the consequences of not developing and funding effective management tools. A lot of the homeowners that we talked to had no idea this was a non-native species, that it was a problem, other than they might not be concerned that they no longer could see the water. But they had no idea that it was having any effect on the functioning of the system. So I think I will leave it at that, and if anyone has questions, I'd be happy to try to address them.
Thanks.